Alright, so now we'll show how to passage cells if you find that your flask is confluent. So what you can do is open it just like we did in the changing media video. Remove the liquid. And we'll want to give it a very quick rinse with PBS. So we just need enough PBS to cover the surface of the flask. Swish it around a little bit. And then remove. And this is just to remove any residual media that's on the flask. Sometimes if you don't do it, the trypsin won't work as well. So now we add a few milliliters of trypsin to our flask. And what we're going to do is we're going to let this sit in the incubator for about 5 minutes, but it will depend somewhat on your cell type that you're using. Okay, so now we have the flask trypsinized, and the cells should be removed from the bottom of the flask. You can tell this by either checking in the microscope, or the flask should actually look a little bit cloudy. So now what we want to do is deactivate the trypsin, because the trypsin actually can damage cells over time. So what we do is we add media in a 2-to-1 ratio to the trypsin. So basically if you had 2 milliliters of trypsin, you would add 4 milliliters of media. So we can add that into our flask, kind of rinse the bottom, mix it with the trypsin. After we add the media to our flask, what we'll want to do is kind of pipette up and down with our serological pipette against the surface of the flask to make sure that the cells are removed. You don't want to do this too aggressively, because what you can do is cause a lot of bubbles to form, and you really don't want that because it'll start making a mess basically. So kind of splash it onto the surface of the flask, make sure all the cells are detached, and then you can take out all of the cells, put them into a fresh tube, and then we can centrifuge these to form a pellet and remove all of the trypsin that was left over from this process. Now we want to take our cell solution and centrifuge it down so that we get a cell pellet. Before we do that, we always want to make a counterbalance, because if you don't, the centrifuge is going to rock back and forth, it'll break the centrifuge, you'll ruin everything. So always have a balance when you use the centrifuge. So we open this up, we can put our tubes on opposite sides of the centrifuge, close, and run at the specified settings. So here's a close-up of balancing the centrifuge. So what you want to do is have things on the opposite side. So you have one on this corner, you're going to want to put your cells on the opposite corner, so that there's a straight line through the center of the centrifuge that connects the two tubes. Alright, so now that our cells are centrifuged, we can spray down the tube, wipe it with a paper towel, and bring it into the hood. So now what we want to do is remove the media from above our pellet, and the pellet should be pretty visible. So when you do this, you can use the aspirating pipette, but you need to be very, very careful. So you need to go slowly so that you don't aspirate the pellet. So we can just gently go, very slowly, and you don't need to remove 100% of the media. You can remove, 99%, and that'll be fine. Remove almost all the media, make sure our pellet's still there, and now we have a pellet that's relatively dry. It doesn't have all of that trypsin on it. The cells, when we add fresh media, will be pretty happy. Okay, so now that we have our dry pellet, we need to add 1 milliliter of fresh media so that we can count our cells and then we can actually dilute the cells or use however much of the cell suspension that we need to repopulate a fresh flask. So we take out our fresh media, we add it to our cells, and then we're going to pipette up and down so that it'll actually disperse the cells throughout the 1 milliliter of media. If you don't do this, then the cell counting is going to be wrong. You'll basically get an uneven distribution, and you might sample the wrong area, and you'll get inaccurate readings. So when you pipette up and down, you shouldn't do it so vigorously that it makes a mess in the tube. You kind of want to gently go up and down. So now we can take cells out of this suspension for counting. Alright, so now that we've counted the amount of cells that are in this 1 milliliter, we need to add the amount of cells that we need for a fresh T25 flask into the flask. Before we do that, we'll want to add in fresh media that we can add the cells to. So what we'll do is we'll add however much media we need for our flask. It'll depend on the size of your flask. So we'll add the media, and then let's say hypothetically we needed 300 microliters of this 1 milliliter of cells to get the desired number of cells for our new flask. So we set our P1000 to 300 microliters. We pipette this up and down again, because any time that you let it sit, it's actually going to start to settle to the bottom. So we pipette this up and down. We take our 300 microliters out, and then we add it to our flask. Generally, I also pipette up and down here to disperse the cells. And now what we should have is a flask with the desired number of cells and fresh media that we can now add to the incubator. So I generally will mix this around to make sure that the cells are spread evenly throughout the flask. The last thing we always want to do is label our flask. So we can label this with our initials, the date, the type of cells, and the passage number.